Today I will be talking to you about mitosis. Mitosis is a type of cell division that generates new cells for growth and repair. It happens in eukaryotic cells. Chromosomes condense in nucleus of cell. DNA condenses by wrapping around cores of histone proteins, forming nucleosomes, which forms chromatin. As cells prepare to divide, chromatin coils up and condenses the chromosome. The replicated chromosomes are called sister chromatids. To prepare for cell division, the cell goes through interphase, which consists of three phases. The gap 1 phase consists of organelles, cytoplasmic components, centrioles in animal cells. All of those are replicated. The synthesis phase, S phase, replicates DNA. The gap 2 phase, the G2 phase, has enzymes needed for process of cell division, which are produced. Eukaryotic cells spend most time in interphase and short time in mitosis dividing. Mitosis results in two daughter cells. It consists of prophase, metaphase, anaphase, and telophase. The first phase, prophase, has chromosomes condensed and appeared as two sister chromatids held together at the centromere. The cytoskeleton disassembles as the spindle begins to form. The nuclear envelope breaks down as centrioles migrate to opposite poles, establishing microtubules and spindle apparatus. At the end of prophase, chromosomes attach by proteins in their centromeres, the kinetochores, to microtubules from each pole, moving chromosomes towards equator of cell. The next phase, metaphase, has the aligned chromosomes at the equator of the cell plate, the metaphase plate, right in the middle. The next phase, anaphase, begins with the degradation of proteins that hold sister chromatids together. These free individuals of chromosomes that begin to separate. The next phase, telophase, creates a cleavage furrow that forms in the center of the cell. This is a constricting belt of actin filaments surrounding the inside of the cell. Chromosomes cluster at opposite poles and begin decondensing as nuclear envelope reforms around. The spindle apparatus disassembles. Microtubules are broken down into tubulin monomers that can be used to form cytoskeleton of daughter cells. And the last phase, cytokinesis, completes the division by completely separating the daughter cells. To make sure that the process occurs accurately, the cell goes through some checkpoints. The G1S checkpoint makes sure that the cell cycle continues or stops the external signals and growth factors. The G2/M checkpoint makes sure that the cells that have completely all three phases of interphase are allowed to begin mitosis. And the last checkpoint, spindle, makes sure that all chromosomes are attached to spindle for anaphase. The factors in the cell cycle regulation contain growth factors, size of cell, and nutritional state of cell. And once these checkpoints are all completed, mitosis can occur.